All right, Bio 30. This presentation is going to be on the function of the neuron. And here I will be talking about how the neuron receives a message, how the neuron is going to pass that message down the length of its axon, and how eventually it will transmit that message onto adjacent neurons as well. This is referred to as an electrochemical message or an electrochemical impulse, and where this term actually comes from. If we take a look at a little bit of the history behind the study of the nervous system, doesn't go back that far, just over 100 years, there was some indication that the message was in fact electrical in nature, meaning that it does involve positive and negative charges and the movement of positive and negative charges. These charges are associated with elements, and of course those are referred to as ions, positively and negatively charged ions. In 1939, a couple of researchers, Cole and Curtis at Columbia University, they took the largest neuron that they could find, largest diameter axon, and that happened to come from the giant squid, the giant Atlantic squid. These axons have a diameter of about half a millimeter in diameter, and that is large enough to take a voltmeter with tiny, tiny little microelectrodes, stick one of those electrodes through the cell membrane and into the cytoplasm, and the other one is on the outside of the cell in what is referred to as the extracellular fluid. When they did this, what they found was there was a voltage difference between the inside and the outside of the cell, and the voltage difference indicated that the interior of the neuron was negatively charged relative to the outside. And this is what we refer to as a polarized membrane or an excitable membrane. Most of the cells in the body, there is virtually no difference between the interior and the exterior, but that is not the case when we are talking about neurons and some other excitable cells like muscle cells. They also discovered, as we'll talk about later, that when the neuron is sending a message, there is a change in the voltage on the inside of the neuron. When they initially stuck the electrode into the neuron was not sending a message and it was said to be at rest. The voltage that they measured at rest is referred to as the resting membrane potential, one of the many terms that you will need to know. Resting membrane potential, the potential or charge difference across the membrane when the neuron is at rest. There are a number of graphs that we will come across that we'll be taking a look at in terms of neuron function. For these, what we will find is that on the x-axis, that is where we have the time in very, very small units of milliseconds. Milli is 1/1000th, so we're talking about 1/1000th of a second, very, very short time frames. On the y-axis, we have the voltage, again, very small units, measured in millivolts. Now, there isn't a huge difference between the interior and the exterior. For most neurons, it is somewhere around minus 70 millivolts, and that is negatively charged on the inside, again, relative to the outside. This is an average number for most neurons and probably not a bad number for you to keep in the back of your mind. A lot of this does have to do with the neuron membrane itself, and if you recall a little bit from Science 10 and Biology 20, the cell membrane, a plasma membrane, phospholipid bilayer is a very, very complex structure. It is made up of two layers, hence the name bilayer of phospholipids. But in this phospholipid bilayer, we also have a number of other structures that can be embedded in it. Included in this are things like cholesterol, proteins, glycoproteins, making it a very, very complex structure. We'll just take a look at a quick little animation here, which is going to take us through some of the basic structures of the cell membrane. So we do see here the phospholipid bilayer. Toward the bottom in blue is the inside of the cell, the systole or the cytoplasm. The top is going to be outside of the cell. Here they're highlighting the individual phospholipids, each one made up of a hydrophilic head and two hydrophobic tails. So the interior of the bilayer is hydrophobic in nature. Now it's showing that we can have a number of other structures associated with it. In this case here we have some proteins. In some cases, they go right through the membrane. In other cases, they're restricted to the inside or the outside. They can shift around. They can move around within the bilayer, and that's where the term fluid comes from the fluid mosaic model. Understanding the processes involved in moving things into and out of the cell is vitally important for the understanding of how a neuron is going to send a message. Again, this should hopefully be a little bit of a review. We can have a number of different processes involved in moving materials into or out of the cell. One of them referred to as diffusion. In this case here, it is showing in fact what is referred to as simple diffusion. For simple diffusion, we have some molecules that are able to move directly through the phospholipid bilayer. In this case here, it looks like it's showing it going from the outside of the cell to the inside of the cell. It could be in either direction. Very, very few molecules are able to pass through the phospholipid bilayer. A couple of examples that come to mind: oxygen, carbon dioxide. Those gases can pass directly through the phospholipid bilayer. Other things can still pass through the bilayer by diffusion, only they have to pass through a special protein. These proteins can be referred to as channels, they can be referred to as gates, but because it is not directly through the phospholipid bilayer, they require some help, and hence the additional term facilitated for this kind of diffusion. Remember, with diffusion, we're always going from where there is a high concentration to where there is a low concentration. No energy is required, so this is passive in nature. We'll take a look at a couple of other quick little animations here. This one is showing the non-gated form that we typically just refer to as a channel, and we'll see that these are very important in the case of the neuron function. Here we do have the phospholipid bilayer, and these red structures are representing a channel in the membrane. Channels are very, very specific, and in this case here, it is only the potassium ion, the positively charged potassium, that can pass through. Again, this is facilitated diffusion. We're going from where there is more at the top to where there is less of it down at the bottom. That is diffusion. The other version here is called gated facilitated diffusion. In this case, the channel is closed. Something does have to open up the gate. That is the hydrogen in this case, allowing the calcium to move through the protein and passing through the cell membrane. Another kind of transport, also very important, now requiring energy. That energy, of course, is going to have to be in the form of ATP. And because it is energy requiring, it is now referred to as active transport. Now we're moving molecules not from where there is a bunch of it from a high concentration to a low concentration, but we're moving it from where there is already a low concentration to where there exists a higher concentration. That does require energy. That is active transport. And we'll take a look at one little Okay, so that was the active transport, the example using the sodium-potassium pump. And as we'll see, that is one of the very important ones in terms of the normal functioning of the neuron. It involves the movement of both potassium and sodium ions against their concentration gradient. It involves the use of ATP, and that's one that we'll come back to and we'll be talking about again. So let's get into what's going on with the neuron itself. So the resting membrane potential, what does that actually look like? Well, there is fluid both inside and outside of the neuron. In this fluid, a whole bunch of things will be dissolved, and many of those things will be various different ions. As we can see here, these are the most important ones that we will be talking about in terms of neuron function. So sodium, potassium, chloride, a little bit calcium ions, and also what are referred to as negatively charged proteins or anions, protein anions. Now, typically what we do find, and these are important details for you to know, is that the concentration of positively charged ions normally equals the number of negatively charged ions on either side of the membrane. The exception is going to be, well, what we are talking about, neurons, and also muscle cells as well. So kind of wordy here. I'll show you a picture on the next slide, but for the neuron, what we do have is a high concentration of sodium and a low concentration of potassium outside of the cell membrane. These square brackets that we see here, those do represent concentration. That is what they're for. So the sodium in square brackets means the sodium concentration. So again, we have a high concentration of sodium outside, a low concentration of potassium on the inside, and it's exactly the opposite inside of the cell where we have more potassium and fewer of the sodium ions. This might make a little bit more sense in terms of where we find the different ions. Numbers, you don't need to know exact numbers, but if we take a look at a couple of the pictures here, if we take a look at the one on the left-hand side, potassium, potassium, even if you take a look at the size that they're using to represent the potassium, the symbol, much smaller on the outside, much bigger on the inside, indicating that at rest, the neuron has more potassium on the inside. Again, exactly the opposite with the sodium. A whole bunch of sodium on the outside relative to the inside. Those are the two major ions that we talk about. We will come across in one or two animations chloride ions, and the A that we see here with a negative charge, that is representing the negatively charged proteins. Picture on the right-hand side, we see three different very important membrane proteins that you will need to know about. This first one that it's showing here is the animation that we did just see, and that is the sodium potassium pump or sodium potassium ATPase. We see two other protein channels here. Kind of deceiving. These channels are not open all of the time, and here it does look like they are open all of the time. But the two that we do see, I guess it is indicated at the top here, the darker purple are potassium channels. They only allow potassium to pass through when they are open. And the other ones, the lighter shaded purple, they are the sodium channels, and they only allow the sodium to pass through when those channels are open. But once again, they are certainly not open all of the time. Here we see these three different channels once again. Remember that these two on the left are channels, so we can only have the movement of the ions going down their concentration gradient. So we typically have, again, more of the sodium on the outside. So you open up a sodium channel, and it's going to rush to the inside of the neuron. Exactly the opposite with the potassium, where we have more potassium on the inside. You open up potassium channels, and it's going to rush to the outside. Far right-hand side of this picture, we do have the sodium potassium pump. So again, what we can see is that it requires ATP. And now it's moving the sodium and potassium in the direction opposite to what they actually want to move in. The sodium does want to move to the inside because there is more of it on the outside. But the sodium potassium pump, it is going to move the sodium back out. It's going to move the potassium back in. Now, if we take a look at this first point at the bottom, it's not an equal movement of the sodium and the potassium. Both of them are positively charged. Both of them have exactly the same charge of +1, but for every 2 potassium that are pumped in, we have 3 sodium that are moving out. So we have this little bit of a disequilibrium It is just positive charges that we're moving, but we're moving more positive charges out for the positive charges that we're moving in. So that is one of the reasons, as we will see, as to why there is a negative charge on the inside of the neuron relative to the outside, is because we are moving more positive charges to the outside. And again, that's due to this pump, the sodium-potassium pump. A couple of other reasons for neurons being negatively charged on the inside, though. Those two channels that we saw, the potassium and the sodium channels, again, they're not open all of the time. If a neuron is not sending a message, the sodium is absolutely not open. The potassium channels, they are what we call a little bit leaky though. So some of that potassium that is pumped to the inside by the sodium-potassium pump, it does leak back out through the potassium channels. So this trickling of more positive charges to the outside, again, contributes to the negative charge on the inside of the neuron. Finally, the third reason for the negative charge on the inside are those negatively charged proteins. Again, we saw this on a previous picture represented like this, and where we had that large concentration was on the inside. These do not move to the outside. They are restricted to the inside of the neuron. So we'll take a look at another little animation. Okay, so what have we accomplished at this point? Well, we haven't sent a message, but now the neuron is ready to send a message. So we refer to it as now being polarized or being charged, and it's ready to go ahead and send its own message. So what I think is kind of a useful analogy is to think about a battery. In a battery, what we really do have is a separation of positive and negative charges. There is a barrier that holds the positive charges on one side and the negative charges on the other side of the battery. They are attracted to each other because they're attracted to each other because they want to move. Here we do have some electrical energy. So all we need to do is connect the two sides of the battery. That will allow the negative charges to move toward the positive terminal, and that allows some work to be done. So separation in the charges, creating a form of electrical energy, tapping into that electrical energy in order to do some work. Well, that's the same thing that we do see. We have a separation of charges, in this case across the cell membrane of the neuron. those charges, they do want to move, but we don't allow them to move. We only allow them to move when the neuron wants to send a message, and that's the form of work that is then being done. And the neat thing about the neuron is that not only is it like a battery, but it is a rechargeable form of the battery. So now finally we can get into what happens when the neuron does in fact receive and send its own message. And this is now what we refer to as an action potential. But again, a whole bunch of other terms that we sort of need to get into first before we can talk about what goes on when the neuron is sending a message. As I mentioned at the beginning, Cole and Curtis, they did find that there was a voltage difference between the inside and the outside. And not only that, when you do stimulate a neuron or when the neuron is sending a message, what we do have is a change in the potential across the membrane, and that is due to the opening and the closing of those different channels that we talked about, the sodium and potassium channels, in a very, very specific sequence. Well, what happens if we take those potassium channels, gated potassium channels, and open them up? Well, remember that there's more potassium on the inside of the neuron, so that means that potassium is going to diffuse out. If potassium is diffusing out, that means that the inside will of course become more negative. Now this is not what happens when a neuron sends a message. In fact, this makes it more difficult for a neuron to send a message, and this is a term that is referred to as hyperpolarization. Initially, the membrane is polarized at rest. If it becomes even more charged in terms of its difference, it is then hyperpolarized. So this, as it says here, is an increase in the magnitude of the membrane potential becoming more negative on the inside. So again, if we take a look at another graph here, we'll take a look at a whole bunch of these graphs. Here we do have, once again, on the x-axis, the time, very, very short durations that we're talking about, voltage on the y-axis. We can see that we have labeled here, straight line going across, the resting membrane potential, and again, that is around minus 70 millivolts. They have identified on here a threshold potential as well. We'll get back to that in a couple of minutes. But the key at this point is we also have labeled here hyperpolarization. So what we can now see is that during hyperpolarization, it's becoming more negative. So for this one here, it looks like we can see maybe this one here going down to about minus 80 millivolts. Again, this is not when a neuron is sending a message. This, in fact, would make it more difficult for a neuron to send a message. When a neuron is sending a message, what we do have is the movement of charges in the opposite direction. So this is referred to as depolarization. Again, another important term for you to know, for you to understand the movement of the ions, and for you to understand what the graph is going to look like when we do have depolarization. So for this one here, if we now open up the sodium channels instead of potassium channels, there is more sodium on the outside of the neuron, so it is going to now diffuse to the inside. Positive charges going to the inside means that it's going to become more positive. So this is, of course, our resting membrane potential once again at about minus 70. Here we now have depolarization, so we have it becoming more positive. Again, when you get depolarization is when you open up the sodium channels, allowing sodium to rush to the inside of the neuron. So when a neuron is stimulated, so whether this is in an actual specimen or whether this is in a lab setting where the neuron has been removed and you're electrically stimulating it, what does happen initially is that the membrane becomes more permeable to sodium. That means that, well, the sodium can move through and it does not initially become more permeable to the potassium. Couple different kinds of sodium channels. At the beginning, we have what are called ligand-gated sodium channels. They're going to open up, and that's going to allow the sodium to rush in when the potassium channels or gates remain closed. So we see then our highly concentrated sodium on the outside is going to pass through the sodium channels. It's going to move to the inside by diffusion and also by charge attraction. That's going to result in a depolarization. So this rapid inflow of the sodium ions causes a charge reversal, and that charge reversal, when the neuron actually sends a message, now it is referred to as an action potential. Charge is completely reversed. It will reach positive values, and this is an average or typical value that we might see with a neuron around positive 35 millivolts. This is what the graph looks like. This is a picture showing a full action potential. So we do have, again, taking a look at the terminology, the resting membrane potential at minus 70 millivolts. We have some depolarization that's taking place here. The rest of the action potential is still depolarization. So everything that I'm showing here with these arrows is all depolarization. But when we have this big peak that we see here, that is referred to as an action potential. So right at the top, that would be what's referred to as the peak of the action potential. And then the neuron does recover. Again, very, very short time periods that we're talking about here. Eventually, it does go back down to the resting state, and again, a bunch of other events and terms that we'll come across that are associated with that. So where this depolarization initially is going to be taking place is at the dendrite end of the neurons. This is where the initial sodium channels are going to open up. Sodium is going to rush in, but the neuron doesn't automatically go ahead and send a message. It needs to make a decision. Is it receiving enough of a stimulus to send its own message? So in order to receive enough of a stimulus and send its own message, there needs to be enough sodium rushing in. There needs to be enough of those ligand-gated sodium channels opening up, allowing sodium to rush in to reach what is referred to as the threshold potential. And again, another number here, somewhere around minus 55 millivolts. Once that is reached, then there's a little bit of a switch that occurs. Once the threshold is reached, now we're going to get a whole bunch of sodium channels that open up. These ones are a little bit different. They're referred to as voltage-gated sodium channels. As soon as you change the voltage to somewhere around minus 50 millivolts, a whole bunch of these voltage-gated sodium channels are going to open up at the same time. And now it's not just a little bit of sodium trickling in, the initial depolarization. Now it's a huge amount of sodium rushing in to get much further and rapid depolarization and an action potential being generated. Now these action potentials, important for you to understand that they are always 100% all or none. There is no such thing as a partial action potential. There is no such thing as a neuron sending a strong message or a weak message. Once that threshold is reached, a message will be sent, and it will always be of exactly the same strength or intensity, hence the term all or none action potential. So a few other graphs that we'll take a look at here. In fact, we've seen much of this already, but now we can take a look at it at the same time and see what's going on with the various different channels, the sodium and potassium channels, and the movement of the ions. So this first one here shows us the resting state. So with the resting state, we're taking a look at number one, and notice that it is both here before the action potential and also at the end after the action potential. So it starts in the resting state and it returns back to the resting state after the action potential. What's going on in the resting state? Sodium channel, for the most part, closed. Potassium channel, still for the most part closed. Now remember, we did say that the potassium channels are a little bit leaky. So whereas the sodium channels, they completely block the movement of sodium when the neuron is at rest, but we can have a little bit of that potassium that does trickle out. Kind of a minor point right now, and they're not showing that with this animation here. When we do go into the depolarization, again, this is initial depolarization at the dendrite end where we will have some, but not all, of the sodium channels that are opening up. So if some of them are opening up, this here is our sodium. Sodium is going to be rushing in, and that's going to make the neuron more positive. So that is depolarization. And we can see on the graph that now it is moving from the minus 70 millivolts up to, well, between minus 50 and minus 60 millivolts. Once we get enough of that depolarization to reach the threshold, we can see that more sodium channels are opening up. We get further depolarization, and then we do get, in fact, an action potential, and this takes us to the peak of the action potential. Kind of going back to the action potential being all or none, this is showing another graph here as well. This graph at the top, the red bar graphs, this is now showing the stimulation of the neuron. So if you give the neuron an electrical shock, it will result in depolarization and possibly an action potential. The first little bit of a shock here, well, it's not a little bit of a shock, it is a threshold level stimulus, which means it is enough of a shock that you're giving the dendrite end of the neuron for enough sodium to rush in to reach threshold and generate, as we can see here, an action potential. So then the question here is, well, what happens if we give it a bigger shock? So a bigger shock that is necessary is called a supra-threshold stimuli. So if we give it a bigger shock than is necessary, the question is, well, do you get a bigger action potential? And the answer is, well, no, it's exactly the same. What if you give it an even bigger shock? Well, still, the action potential is the same. So the action potential is always going to be all or none. You either get it or you don't reach threshold, and you do not get the action potential. So now we need to recover, the neuron needs to recover, and we need to get back to the resting state. So once the voltage inside the neuron becomes positive, those sodium channels that opened, they're now going to close and will no longer have any more inflow of the sodium. But as soon as they close, the potassium channels are now going to open. Remember, there's more potassium on the inside of the neuron, so it's going to move to the outside. So now we have positive charges that are going out. If positive charges are going out, that means the voltage is going to become more negative once again. So going in the opposite direction of the depolarization is what is referred to as repolarization, and that is due to the opening now of potassium channels. So back to these pictures here. We see that number 4, the sodium channels, they are closed. Not only are they closed, I'm not sure if you notice this here, but they're in kind of a different state than what we saw initially. They're showing here what's referred to as an inactivation loop. So these sodium channels, they are not only closed, but what we'll see a little bit later is they're kind of inactivated. They can't be opened for a short period of time. But here, of course, we do have the potassium channels that are now open. They open up, potassium rushes out, and that's why we see this taking place. So now it's going back to the more negative values, and that is our repolarization. And the final one that we have here, now they're not showing all of this, but they are showing what's referred to as an undershoot. It is going down below the resting membrane potential, so this is also a hyperpolarization that we see here. Undershoot, hyperpolarization, after hyperpolarization. This is after the repolarization that we're also seeing this hyperpolarization here. Now there are some other things taking place at the same time to eventually get the neuron back to the resting state. But this is all of the events that are occurring in terms of the sodium and potassium channels during a full action potential. But now we do have a little bit of a problem. The sodium rushed in, the potassium rushed out. The voltage might be restored, but the ions are in the wrong location. So we've talked about the sodium channel. We've talked about the potassium channel, but now we need to go back to that sodium-potassium pump. So we need to take that sodium and pump it out, and we need to take the potassium and we need to pump it back in. And that again is the function of the sodium-potassium pump. So not only does it generate the initial resting potential, it also gets the neuron back to the resting membrane potential after there is an action potential. Let's take a look at all of this together then. So the picture at the upper left-hand side, we have the resting membrane potential here. Again, you should know the term that goes along with the graph, that goes along with the channels, and the ions. So what's taking place here? It's the resting membrane potential. It's at minus 70 millivolts. Sodium channels are closed. Sodium is not moving through the membrane. Potassium channels are closed for the most part, and potassium is not moving through the membrane. We jump over to number 2. We have depolarization. It doesn't show the threshold potential here, but depolarization with the sodium channels now open. Potassium channels are still closed, and we can see that the voltage is becoming now more positive. Jumping down to number 3 at the lower left-hand side. This one here, we have reached the peak of the action potential. Now we can see that the sodium channel that was open is now closed, and the potassium channel is now going to be opening up. And now we have not depolarization, but repolarization. And finally, really just back to the beginning, again, we don't have the sodium-potassium pump here. That would have to be taking place, but we still have restored the original resting membrane conditions after the repolarization is complete. A little animation here, hopefully. Looks like this one is just showing a whole bunch of consecutive action potentials, one right after the other. And you can see here, in fact, that they are all the same size, same shape, same size. And again, that's the idea behind the term all or none in reference to the action potential. So here it is, most of what we talked about, voltages, terminology, what goes along with that, of course, the channels, are they opening, are they closing, which ones are opening, which ones are closing, the ions, which ones are moving, in which direction are they moving in. So we have our resting membrane potential that we're starting with the minus 70 millivolts. That's both at the beginning and at the end of the action potential. It's identified the stimulus that's taking place at the dendrite end. Now these ones here, these failed initiations, that's if there's not enough of a stimulation for sufficient ligand-gated sodium channels to open up, insufficient sodium rushing in to reach the threshold, and the neuron is just kind of saying, well, no, I'm not going to send a message, so it goes back down to the resting state. Once the threshold is reached, though, now many voltage-gated sodium channels will open up. We get further depolarization as the sodium is going to rush in, eventually reaching a full action potential. And the peak of the action potential, here it's showing it at around plus 40 millivolts. Once we reach that peak, sodium channels close, potassium channels open, potassium rushes out, we get repolarization. Eventually, all of this in here, we've seen a couple of different terms for this, the undershoot, hyperpolarization. We have the sodium-potassium pump that's working away, but now we have another term here as well, something referred to as the refractory period. The neuron does have to recover, and you can think of it as it needs to take a little bit of a rest, needs a little bit of time in order to restore those initial conditions. So refractory period following an action potential, neurons cannot be activated again until the resting membrane conditions are restored. The period of time is very, very short. but it's necessary before a subsequent action potential can occur. So 1 to 10 milliseconds, the duration of the refractory period. And again, this picture here, we can very nicely see the channels, sodium-potassium channels, and when they're opening, when they're closing at different stages of the action potential. Two different refractory periods you will not need to know for purposes of the diploma exam, these two different ones, but they are referred to as the absolute and the relative refractory periods. Absolute refractory period, it's absolutely impossible to get another action potential because the sodium channels simply cannot be open. Not only are they closed, they're temporarily inactivated. During the relative refractory period, it is possible to get a second action potential. It's due to now the potassium channels that are unable to open, but sodium channels can still open. A second action potential is possible, but it does require a much greater stimulus, and the threshold is typically higher now in order to get that second action potential if the neuron has not fully recovered. And this just kind of shows us where the refractory periods fit in. So from the peak of the action potential to the end of the hyperpolarization, and again, notice here they're calling it the after hyperpolarization. That is the entire duration of the refractory period. Again, a couple of other graphs here. At the bottom, we do have the action potentials. At the top in red, once again, we do have the stimulus. So this one here is showing that we're starting initially with more than sufficient stimulus to reach the threshold, so we do get our action potential. Then we wait a period of time and we give it not only another stimulus, but a continuous sustained suprathreshold level stimulus, more than enough to generate an action potential. So what we get is action potential after action potential taking place here. But there's also a space between the action potentials. So that space between the action potentials does represent the refractory period, and in fact the absolute refractory period. You cannot bring these peaks any closer together. That is the absolute minimum amount of time that is required for the neuron to recover. Just a couple of cool things, I guess, about neurons and the function of neurons. Um, Dealing with pain and pain medication, something very, very tricky, I guess, for medical doctors, and something that a lot of research is going into in terms of coming up with better painkillers. Opiates, things like morphine, they're kind of effective, it appears, in the short term, but can lead to obviously some long-term effects, effects associated with things like tolerance and addiction to those kind of drugs. There are some local or topical anesthetics as well. The one that I'm showing here is for lidocaine, and this is kind of a neat one. So this topical anesthetic is one that prevents depolarization, and the way that it does that is it prevents the opening of the sodium channels. So the left-hand picture here just kind of showing what one of these sodium channels does look like, and in fact it's a very, very complex structure that we do have here, complex protein. But the local anesthetics, and I'm not going to read through this whole description, but these are showing that the local anesthetics, they're going to block this sodium channel. That's going to prevent the sodium from rushing in. If you prevent the sodium from rushing in, and we're talking about a sensory neuron that's sending messages to the pain processing region of your brain, well, you're going to be blocking that message from reaching your brain. And that more or less is how the local anesthetics like the lidocaine, how they're going to be functioning. I'm just going to let you read through this. I'm not going to go through all the details of this, just some kind of neat things on these pain receptors, these nociceptors, and some additional sort of drugs that they're coming across from various different sources. Kind of neat where they can get some of these different medications from snakes, as we can see here, from cone snails, and from centipedes as well. Each one is going to target different kinds of sodium channels, so each one may have different applications for different components of medicine. So the final part of this rather lengthy presentation, now what we've done is we've really generated an action potential at the dendrite end of the neuron, but we haven't really passed it on through the length of the dendrite to the cell body, through the cell body to the axon, and down along the length of the axon to the axon terminal. So how do you actually do that? I mentioned those different kinds of sodium channels, the ligand-gated and the voltage-gated sodium channels. So it's kind of the voltage-gated sodium channels that we're going to focus on here. So voltage-gated sodium channels, they're triggered to open by a change in voltage across the membrane. So if you change the voltage in one region, it will open up voltage-gated sodium channels in the next adjacent region. This clip here, I don't think I'll play this clip. You can play it in D2L. It's just titled How Neurons Work, and it's a couple of minutes in length, so I'm not going to play that right now for you, but you can take a look at that on your own. This one here is showing us a longitudinal section through the axon portion of the neuron. And what it's showing at the top is three different regions. The beginning of the axon, region A, the middle portion B, and closer to the axon terminal, the C region. So what we can see here, what this is trying to show with the graph anyway, is that an action potential is not taking place along the length of the entire neuron. It's taking place in one region where we have depolarization. We get an action potential in that region, and then eventually the message is passed on to the next region. So in the middle, now what we can see is that region A, it is now recovered. We don't have the action potential there anymore. Now the action potential is right in the middle, and we can see with the graph. And then finally, for the third segment at the bottom, it shifted along to section C, and that's where we have the depolarization. So this is what we refer to as impulse conduction or propagation of the action potential. This one here again, a longitudinal section through the axon, and just showing a little bit more clearly what is going on with the ions. So where we have the action potential, we have the sodium channels that are open, we have sodium that is rushing in. Now we shift along to the next segment. That is now where the sodium channels are open and sodium is rushing in. But what about where the message just came from? So the message is traveling from left to right. Where the message has just come from, well now this region right here is in repolarization. So sodium channels are closed and now it's potassium channels that are closed. Or sorry, potassium channels that are open. And then we just shift along to the next region. So where the message is coming from, all of this region that we see here, it's undergoing repolarization, it's undergoing hyperpolarization, and it is in refractory. That region is in refractory. And it's for that reason that we cannot have a message that is going to go back and travel in the same direction that it has come from. Once again, with this picture here, the message is going from left to right. We can see right in the middle here, that is where the sodium channels are open. Where the message has come from, these sodium channels are inactivated. Where the message is going to, the sodium channels are closed but not inactivated. There's a little bit of a difference. Where they are closed, they can open. And in fact, that's what we see in the next segment, is they have opened up. Where the message has come from, though, they are inactivated. Those sodium channels cannot be open, so the message cannot possibly travel in the direction that it has come from. And that's just a little bit more of this picture here showing that we only have the action potential traveling in one direction. Okay, hopefully this is all starting to slowly come together with the ion channels and the movements and the different terminology. Final thing, speed of conduction. How fast is the message actually traveling down the length of the neuron? Well, we're not quite talking about the speed of light. We're not talking about an electrical message traveling down a copper wire. It's somewhat slower than that, but still pretty darn speedy. So here, the red bars, what they're showing is a couple of different nerve fibers and the speed that it's traveling at. Unfortunately here, it's in miles per hour that it's showing instead of kilometers per hour. But this one here, we're talking about, well, 250 miles per hour. We're talking 400 kilometers per hour. So fairly speedy in terms of how fast a message can be sent. Two important things that you do need to know about that determine how fast a message is going to be sent. Remember that squid axon, half a millimeter or more than that in diameter. That's one way that an organism can increase how fast a message is traveling down the length of the neuron is to increase its size. And by size, we're really talking about the cross-sectional diameter. And this decreases the resistance and increases how fast that message is being sent. So what it comes down to, the fatter the neuron, the faster the message is going to be sent. So this is showing here the diameter, and here it's measuring in micrometers. So micrometer is 1/1,000,000th of a meter. So we're talking, again, very, very small diameters here. But we can see a fairly clear trend that as we increase the diameter of the neuron, it increases the speed, and pretty much a linear relationship that we see here. The second way that a message can travel faster down the length of the neuron is something that we have seen before. That is the myelin sheath that's wrapped around the axon. So the myelin sheath does come from a very special kind of glial cell called Schwann cells. These Schwann cells are in the peripheral nervous system. If it's in the central nervous system, there is still some myelination in some regions of the brain and the spinal cord, and that is by a cell type that's referred to as oligodendrocytes. But at any rate, this myelination is a fatty material, and it insulates the cell against the movement of ions. So what that does mean is that we cannot have sodium, potassium, or any charged ions that are crossing the cell membrane where you have the myelin sheath. And in fact, there are no sodium channels underneath the myelin sheath. So what happens is you still need to get an action potential. You still need to have the message that's traveling down the length of the neuron. But where you can have that action potential is only in the gaps between the myelin sheath. These gaps, very, very small distance that we're talking about, these gaps again are called the nodes of Ranvier, and that is the location where you get the action potential. So if you get an action potential at this node of Ranvier right here, it's going to open up the next adjacent voltage-gated sodium channels, which just happen to be way over here. And then the next one's way over here. So this speeds up how fast the message can be sent in a process referred to as saltatory conduction. So let's take a look at, well, I think one final animation here. This one here is showing an unmyelinated neuron, where we can see that all along the length we do have to have sodium channels that are opening up, sodium that are rushing in, and an action potential at every single location along the length of the neuron and along the length of the axon. When we have saltatory conduction, we can see it's considerably faster due to this jumping from node to node, bypassing the region where we do have the myelin sheath.